Welcome to my lab. This is a standard plant molecular biology lab. If you look around, most of the people milling about are graduate students and a research technician and undergraduate students. And together we are all interested in addressing the question of how plants respond to environmental signals. And some of the signals that we think about in here are drought, salt, cold, and even things like low nutrients. And you probably didn't know that plants, and they manufacture their own food, but they also get into some situations where nutrients are low. Specifically, we're interested in the molecules that plants use when they're sensing and responding to the environment. So plants are actually using specific molecules to perceive the environment around them. This whole process is called signal transduction. And to tell you more about that, I really want to show you. So when we think about plants responding to the environment, we usually think of the whole organism, and I've drawn a plant here. And we think about them responding to things like light, and I think I mentioned cold and salt and drought stress. So there are lots of different signals. But what I want you to do is to think about the cells inside the organism. So I'm going to draw a cell for you, shown here, and I want to talk about how those signals get inside the cell. And so I'm just going to draw an arrow from the various signals, and this is really the process of signal transduction. So we're going to transduce that signal to the inside of the cell. And so basically what we know is that there are specific types of things inside the cell that help amplify the signal. And then we just really think about a series of arrows that all end up causing some final biological response. So this is a signal transduction pathway, or just a pathway for short. So one way to understand this signal transduction pathway is to take away one of these arrows. And my lab has been interested in a group of genes that we think are important for this process. And so to study the pathway, what we've done is made or identified a group of mutants. And the way that you can think about a mutant is it just takes away one of these arrows. You lose the function of that arrow. And so one of these arrows is something we call P80. And P80 is a gene that encodes a protein, and the size of it is about 80, and so that's why we named it P80. And P80 is very interesting. When we look at the mutant of P80, it itself differs depending on the environmental conditions. And I would like to show you that. So we're now in a growth room, and what you see are a series of growth chambers. They're light and humidity and temperature controlled. They're called reach-in chambers because you can just reach in and get your plants. And so what I'm holding is the P80 mutant, and then a plant that I'm going to compare it to called a wild-type plant. This plant has a normal P80 gene functioning in it. So if we have a look at these plants, so this is the P80 mutant, and this is the wild-type plant. And grown under these conditions, for the most part, they look very similar. If you look very closely, it's hard to see, but if you look closely, you may notice that the leaves on the wild type can be a little bit more elongated, and on the P80 mutant, they can be a little bit more round. So the take-home message is, under these conditions, P80 looks very similar to the wild type plant. So this is another set of P80 mutant plants. And what you'll notice right away is they look very different. And in particular, you'll notice that some of the leaves look like they're dying. That's called senescence, and it's happening very early. These plants also flowered early, and you can see the flowers at the top. And overall, they're very small for Arabidopsis. So now that we're back in the lab, I would remind you that when you're growing P80 in the classroom, P80 mutants, you may see different phenotypes. And part of your challenge is to help us figure out what some of the factors involved are. So if you can tightly control your experiments, you can help us understand how P80 helps regulate signal transduction in plants. I look forward to hearing about how P80 grows in your classroom and what you find out.